All right, well, thanks everyone for joining the first of our FIA soil sessions today. I'm Melissa Pastore and I'll be moderating this session. Today, we'll hear from presenters from a variety of institutions who have used FIA soil data in their research on diverse topics like carbon cycling, microbial communities, fire disturbance, and innovations in soil measurements. Just a reminder that each speaker has 15 minutes, including time for questions at the end. So to stay on schedule, we'll have to stop presentations that run over time in fairness to everyone. But we will capture any questions that are left in the chat and try to move those over into Hoopa for later. And so I will be our first speaker for today. So I just joined FIA about a year and a half ago as a research ecologist, and one of my priorities has been supporting the soil monitoring program, which is part of the NFI, or Nationwide Forest Inventory carried out by FIA. And I think most people are less familiar with FIA's soil assessment than the tree assessment. So my goal today is to introduce FIA's soil monitoring program and set the stage for the other great talks we have lined up for our 2 sessions. I'll provide an overview and status update of the soil program, briefly explore a few applications of the data, and highlight a few potential improvements and future directions. What is now the soil monitoring component of FIA's Nationwide Forest Inventory was originally the soil indicator of the Environmental Monitoring and Assessment Program, an EPA research program that developed tools for monitoring national ecological resources. The soil indicator, along with other forest health indicators, were folded into the Forest Health Monitoring, or FHM program, with its formation in the early 90s due to increasing concerns about forest health and air pollution. While FHM was conducting forest health assessments, FIA at the time was conducting forest inventories primarily to report on the trends of timber and woodland species. But after the 1998 Farm Bill, the FHM and FIA plot networks were integrated, and beginning in 2001, the soil indicator was implemented as part of the FIA Forest Ecosystem Health Indicators program within the NFI. Since that integration, a subset of FIA's standard phase 2 or P2 plots, shown here colored by region, have been designated for measuring different forest ecosystem health attributes, including soil characteristics. This means that FIA provides co-located soil, tree, and site data that allow for the exploration of relationships between above and below ground factors. The size of the soil subset now varies among regions, but includes at least 1/16 of the forested P2 plots shown here, and I'll talk more about that in a bit. The soil variables monitored are diverse, ranging from metrics of soil erosion and compaction, nutrient and water availability, the accumulation of toxic substances and soil acidification, and other metrics that help to inform forest health more broadly. The percent of bare soil in each subplot is used to model erosion potential by combining it with precipitation and other data. The percent of soil compaction is measured on each subplot, and the type of compaction is assessed, like a rutted or compacted trail. Soil samples are collected, and some physical properties are measured in the field in association with sampling, like soil texture, the thickness of the litter layer, and full organic horizon, termed the forest floor in FIA. Depth to a restrictive horizon is also recorded if there is one. Lab measurements include bulk density, carbon and nitrogen concentrations, coarse fragment content, water content, pH, extractable phosphorus and sulfur, exchangeable cations, and trace metals. This diagram on the left is a cross-section of the soil horizon showing FIA's soil sampling and depth measurements. Overall, FIA collects 3 samples of the forest floor per plot, the brown and black layers in the diagram, and 1 sample of mineral soil split into 2 depth intervals, 0 to 10 and 10 to 20 centimeters. Those samples are then sent to labs where they're analyzed, and then any leftover soil is archived. This is a diagram of an FIA plot. The forest floor samples are collected along sampling lines adjacent to subplots 2, 3, and 4, and the mineral soil samples are only collected adjacent to subplot 2. Sampling sites vary on subsequent visits, alternating on opposite sides of point 1. There are 3 meters between each sampling point because we don't want to resample a spot where we removed and disturbed soil in a previous year. So far, there are over 10,000 plots that have been visited for soil measurements, and of those, 2,360 had at least 2 visits for soil sampling. Those remeasurements are colored orange and red here, so you can see that they're largely in the NRS and RMRS regions. And as more remeasurement data become available, it will also be possible to examine trends in soil properties in concert with changes in forest productivity and composition. Throughout the long history of FIA soil inventory, there have been some differences among regions in terms of sampling across space and time as funding and priorities have shifted. For example, the Rocky Mountain Research Station has been doing soils continuously since 2000 and moved to an all phase 2 sampling scheme with a long-term schedule of at least 1 soil visit on all field visited forested P2 plots, whereas the NRS had a break in sampling for a few years and is measuring 1/8 of P2 plots by alternating between 2 1/16 subsets. The Southern Research Station primarily sampled soils in the early 2000s, but has since stopped. The Pacific Northwest Research Station now only samples soils in interior Alaska and Hawaii, which use different protocols than other regions because of unique soils and challenges. So there are some sampling scheme and protocol deviations from what I described on the previous slides. And they're collecting soils from all field visited forested P2 plots. And if you're interested in hearing more about Alaska and Hawaii soils, definitely stick around for a couple of talks coming up. The soil data generated so far have been used for a variety of reporting and research efforts. This is a list of some relatively recent studies that I'm happy to share. If you want to take a closer look, just give me a shout or hit pause if you're watching the recording, and you'll hear about some of this work today if you stick around. The applications are really diverse. One key area of interest is the spatial distribution of soil carbon content. This is a map of mineral soil carbon stocks to a depth of 20 centimeters on forest land using the most recent plot measurements of soil carbon concentration, bulk density, and coarse fragment content. And these direct soil measurements are now being used to predict soil carbon stocks across the entire FIA network of forested plots. And I won't say anything more about that because Grant Donkey and Ashley Lang are going to share more about these carbon estimates in our second session. The soil data can also be used to assess soil quality. Several years ago, FIA developed a soil quality index using 19 measured physical and chemical properties of forest soils, with higher SQIs indicating greater relative soil quality. Early results using this index suggest that overall, soil quality has remained relatively stable on plots with at least 2 measurements. And this is a concept we're revisiting, so we're interested to hear thoughts on SQI metrics and when they're useful. I'm planning a new study with Randy Kolka and John Shaw to measure changes in soil mercury content by leveraging the archived forest floor and soil samples. There was an initial investigation done in the early 2000s looking at mercury concentrations in forest floor material. And now that about 20 years have gone by, we want to build on that work to see whether anything has changed, look across a broader region, and assess mercury content in mineral soil as well. Part of what I'm working on in my new role, in collaboration with others across the regions, is identifying opportunities to improve and build upon FIA's soil program. So I've highlighted just a few here. In terms of the protocol, FIA collects mineral soil down to 20 centimeters, but there's evidence to suggest that increasing the depth of soil sampling may be warranted. Litter and the underlying duff are currently collected together, but it could be useful to determine carbon stocks in each of these organic layers separately. They may respond differently to disturbances and agents of change. And if you stay for the next two talks, you'll hear more about how FIA data are being used to explore the factors that influence soil carbon. FIA has a limited ability to capture soil variation at small scales, and because we only take one mineral soil sample per plot, it's difficult to know at the plot level whether we're really tracking changes over time after a remeasurement versus capturing spatial variability because we're sampling a slightly different location in the plot. So that's where having a high sample size is important. The Rocky Mountain Research Station is running a pilot project, taking 3 mineral soil cores per plot to help characterize within plot variation. And we're thinking about this at the NRS as well. Currently, we don't make any measurements related to soil microbial communities as part of the national inventory. Changes in microbial abundance, composition, and activity can signal shifts in soil health and function that might not be apparent from physical or chemical analyses alone. And a study is underway to investigate soil biota on archive samples, which you'll hear more about today if you stick around. We could consider more advanced soil texture measurements. Currently, broad texture classes are assigned by feel. However, the Rocky Mountain Research Station is now analyzing soil texture for their new and archived samples using laser particle analysis, which could potentially be incorporated into standard FIA soil procedures. While there's always room to improve, FIA's soil program has been successful and is doing a lot of things well. Some of the key strengths of the soil inventory are that it allows calculation of statistically robust estimates of soil attributes with associated estimates of uncertainty. It's comprehensive where implemented, and that all ownerships, ecoregions, forest types, and forest soil types are included. It provides the means to track changes in soil attributes over time. It can be integrated with other FIA forest data from the P2 plots, making it possible to look at several ecosystem components together and in relation to things like disturbance or management treatments. Finally, FIA's estimates of soil organic carbon stocks are now incorporating real physical measurements from field collected soils. So overall, since starting this position, it's been exciting to learn what FIA's soil inventory has accomplished so far. And I see some nice opportunities for it to keep evolving. And we'd love to hear what ideas and needs you all have as well. So thank you very much for listening. And I'm happy to hear your thoughts and try to answer any questions you have in the next 1 to 2 minutes. All right, I see a couple in the chat. I don't know if I don't see any hands raised. Do I think that FIA will ever have the resources to go deeper than 20 centimeters? It's a really good question. I certainly don't have the answer to that. I guess I'd say I try to just remain optimistic. But I think there's also a question of if we had more funding, what would be the first thing that we would do? Would it be deeper sampling, or would it be more plots or more within plot? And so I think it's great that we have this national team now to help sort of figure out what those priorities are, but who knows what the future will hold. And any other questions, I'll be happy to answer those in the chat or through Hoover later, but it is 2:30. And so I want to turn now to our next presenter. Next, we'll be hearing from Noel, a postdoc with USGS, who will present about the drivers of forest floor and mineral soil carbon pools in northeastern US forests.
Awesome. Thank you for the introduction, Melissa. I'm sharing my screen now. Hopefully you all can see these slides. All right. Yeah, as Melissa mentioned, my name is Adam. Postdoc right now with the USGS Southwest Biological Science Center. And I'm presenting today on what drives carbon pools in northeastern US forests. I worked on this project when I was in graduate school at University of Vermont, and had an awesome batch of co-authors and researchers thinking about this project with me, spread across University of Vermont, University of Michigan, University of Quebec, Montreal, and then of course the Forest Service. So today I'm going to be sharing just some brief background, why does soil carbon matter, some short and sweet methods, some results from our models about what drives mineral soil carbon and what drives forest floor carbon, and then thinking about what implications might be for carbon pools in northeastern forests. So we're all aware of the current ongoing climate crisis and efforts to try and curb CO2 emissions into the atmosphere. Over the past presidential administration, we've had several executive orders that try and direct management agencies to addressing this issue, which comes in forms of huge national scale carbon sequestration inventories, trying to track how carbon is stored in our forests and our landscape. And the forest inventory analysis is really a great inventory system and resource to be able to do this kind of research and track carbon in the landscape. So the forest inventory and analysis has already helped us in the past. It's helped identify that the eastern US and the northeastern US in particular have been carbon sinks over the past couple decades. And this is really due to kind of the forests regrowing from previous mass deforestation as we colonized and settled across this landscape. So above ground biomass holding carbon in trees is great, but soils are an even better reservoir as carbon makes its way from tree biomass into the soils. It can hold more in terms of just a bulk amount, and it also holds on to carbon much longer. So we assume that forests, as they gather and accrue forest or CO2, that there has to be some pipeline between forest carbon, the litter as it meets the soil surface, and then the underlying mineral soil carbon. And carbon is entering the soil system in a couple different forms: via leaf litter, via root exudates, or via some larger biomass inputs. And of course, if forests are gathering this carbon, we think that some characteristics of the forest must have an influence on carbon stocks in these sites. So tree traits like wood density or leaf nutrients or stress tolerances might affect the way that carbon is gathered and makes its way into the soil system. And of course, we know that there are many other factors at play in our forests, things like climate, topography, and then other soil characteristics. And to make it even more complicated, there are plenty of interactions between these factors that all influence the way that carbon moves through these systems. So with that in mind, really the main goal of this project was to try and disentangle some of these drivers and get at what is driving these forest carbon pools in the Northeast. And I'm really stoked to be presenting here at the symposium because the forest inventory analysis data set is really one of the few that can answer these questions at this really broad scale, with the paired above ground tree observations and measurements paired directly with site soil characteristics. It's a really cool resource that we have available. So our hypotheses with these projects are that the forest floor carbon pool is going to be influenced by climate conditions, forest characteristics, that the underlying mineral soil carbon pool will be more influenced by soil conditions, precipitation, or topography, and that these carbon pools are linked, that the forest floor carbon pool must have some influence on the mineral soil carbon pool. So to try and answer these questions, we used FIA plots across the Northeast, 542 sites from Ohio to Maine, and then we supplemented the FIA data with other metrics, things like measured soil texture, so like clay, silt, sand percent, and then other forest traits, climate conditions, and topography. And then we modeled all of these predictor variable effects on the carbon pools using boosted regression trees, which is just an ensemble machine learning method that tries to create a model that best predicts your response variable. I'd be happy to go over more methods afterwards if folks have any questions. And so in this project, we created several models with different groups of predictors to try and predict our forest floor or mineral soil carbon pools. Here's kind of a short list of those predictors. We split them into two broad groups, abiotic and biotic. So in abiotic, again, things like climate, topography, and soil characteristics, soil nutrients that kind of represent fertility, texture, and percent of these texture classes, and then mineral contents, so iron, aluminum, and manganese oxides, which are kind of these amorphous soil minerals that we think have something to do with carbon storage. Biotic characteristics, stand age, density, a portion of that stand that's conifer, some diversity metrics, and then traits, which are measured at the stand scale as either a community mean or a diversity metric of that trait. So I'll go over results for our project really in just three main categories. Thinking about model performance, how well does that model do and how does it change with or without certain predictors? We're going to look at the important variables in each of these models and how they influence the carbon pool, and then think about the response shape of mineral soil carbon or forest floor carbon to the different predictors in the model. So I'll just, big overview of results right now, and I'll slowly walk through these model results. These models are all testing mineral soil carbon as a response. And we made several different versions of these models. So one containing all the variables, which are listed here on the right side of the screen for reference, models that contain just biotic variables, or ones with just abiotic variables. And you can see from this list that models with all the variables available were the best performance, explained about 80% of the deviance in mineral soil carbon. So among these models that we tested with all variables, we can see the relative influence of variables that were included in the model. These are just the top 10 from the model. And you can see that soil nitrogen had the highest influence, followed by several other soil characteristics, soil iron, aluminum, potassium, pH, manganese, and calcium. And you can see that's really soil characteristics that have the highest influence in this model. Next thing we wanted to do is test this model without soil nitrogen. And we did this because it's really hard to discern what soil nitrogen is doing in the model. Is it a measure of site fertility that was already present at the site? Or is it kind of a measure of nitrogen contributions from organic matter cycling at the site? Kind of hard to disentangle what might be happening with soil nitrogen here. So we took it out of the model to see what would happen. The performance was worse. We had more mean residual deviance left over. And you can see some of these highest performing variables are fairly similar. There's still these soil characteristics. Next, we wanted to test this model, and again, without soil nitrogen, but we also omitted forest floor carbon this time. And that's really the variable that we're interested in here, trying to see if forest floor carbon has an influence on mineral soil carbon. You can see that the model performed better. There's less residual deviance when we take forest floor carbon out of the model, which is kind of interesting. And then the highest performing variables remain these soil characteristics. So what did we learn from this iteration using all variables in our models? Well, including forest floor carbon does not improve the model, and that when we're testing all these variables, that abiotic variables are more influential than biotic variables. All right, so if abiotic variables are more influential, what happens when we test models with just the biotic variables? Right from the outset, we can see that there is much more mean residual deviance. The models are not performing as well. And then looking at these highest performing variables, we see forest floor carbon as a top performer, and then followed by several other metrics that kind of have relationships with deciduous or coniferous forest composition. So again, we tested this model, removing forest floor carbon, and we again see that model performance was better without forest floor carbon included in the model. And then we can see some of these other variables in the lower left take up some of that influence explained. Relative density is now the highest performing or highest influential variable, followed by forest floor nitrogen. And so what do we learn from these models? Again, we see that including forest floor carbon does not improve the model, in that traits that are associated with conifer and deciduous composition are important here. And last up, testing mineral soil carbon. We tested some models looking at just abiotic variables. Mean residual deviance, again, this model does not perform as well as one with all variables, but it still performs better than the biotic model. And we can see these variables in the lower left, things like aluminum, iron, and manganese all have the highest relative influence. So the first time we went through with this project, we tried to use, hand classified texture as a predictor, which didn't work too well. When we submitted this project for reviews, reviewers did not like that we didn't have clay or sand or silt percent We came back through, included those variables, and I'm just explaining all of that to say that this next test, we omitted soil aluminum and iron to kind of get at this idea that, well, these are the variables that are explaining the most deviance here. They must be important. What happens if we remove them? Do we get silt or clay boosting up in importance? And we did not see that, which kind of leads us to this kind of new proposed storage mechanism. Iron, aluminum, and manganese oxides are all highly important in our models. And we're kind of seeing that the oxides in these kind of models are more important than soil texture and driving storage, which is kind of cool to see. I know that we're all aware that clay and silt content, these charged particles can drive soil carbon storage, but it was cool to see that these other soil oxides may have a really large influence that we have not observed before. So again, kind of a recap of these mineral soil carbon models. Model with all variables performed the best. Abiotic variables ended up being more important in these models, and especially iron, aluminum, manganese oxides. And then biotic variables were less important, but they still contributed to deviance explanation. And I'm going to go through just one quick slide looking at our forest floor carbon models, where we tested a batch of all variables and a batch of just biotic variables. And in this case, you can see that the models with just biotic variables ended up performing better than a model with all variables included. And some of the top variables from these models in predicting forest floor carbon were things like forest floor nitrogen, pH, soil aluminum, and leaf nitrogen, relative density, and mean annual temp. And so thinking about these models for forest floor carbon, we saw that biotic model is better than models including all variables, and that some similar variables predict forest floor carbon as we saw in mineral soil carbon models. So this is the last set of results I'll be looking at, or we'll be sharing today, just looking at these important predictors from the mineral soil carbon models and thinking about the response of carbon to these predictors. So this top row are our soil storage metrics, so iron, aluminum, and manganese. And you can see as you go left to right on the x-axis, as we increase in soil, iron, or aluminum, the predicted response of carbon is to increase. This next row is kind of looking at soil nutrients that might be more associated with site fertility, so potassium, calcium, forest floor nitrogen. Again, you can see some effects of increasing potassium, predicted increase in carbon, same with forest floor nitrogen. And then this last row of plots is just looking at some of these important forest composition metrics. So the community weighted mean of leaf longevity, conifer basal area at a site, or relative density. And you can see some of these effects are a little bit weaker across their range, but they still do suggest that these forest characteristics are having an influence on soil carbon at these sites. And then here is just kind of the shape of forest floor carbon, even though it was a poor predictor. So overview of this project, we saw that including both abiotic and biotic variables in a model is best at predicting mineral soil carbon. Site conditions alone were better predictors than forest composition alone, and forest metrics that are associated with conifer and deciduous trees are important in varying effect. And not really sure how to interpret the effect or lack of effect of forest floor carbon. And so what does this mean for carbon sequestration efforts? It might mean that the site conditions of a location are more important than the forest at the location. It may also be, if we're able to identify good relationships between places with high carbon potential, we may think about managing those places for carbon storage differently than we would manage other places. And this last slide I have is just, what kind of data could potentially improve this project. Melissa had pointed out some of the stuff in her presentation, possibly breaking up carbon into different types, thinking about root carbon inputs or root traits. Deeper soil data would be awesome, and actual texture percent data from the FIA sites directly would be great. FIA might be able to add it, but I've sampled among the FIA sampling strategy before, and I understand how much effort it is to make any adjustments. And I would love to take questions, but I think I might be out of time. So thank you all for listening. Really happy to share this project with you all.
Thanks so much, Adam. We are out of time for questions. It looks like you have some messages in the chat that you can take a look at and Again, we will move these over to Huva as well. So our next speaker is Haley Peter-Contesse from the University of Hawaii at Manoa, who will present insights into soil carbon spatial drivers and distributions across Hawaii's forests.
Thank you for the intro, Melissa. I'm just going to share real quick. Again, aloha, and my name is Haley Peter-Contesse. I'm a postdoctoral researcher at University of Hawaii at Manoa. And today I'm going to share some insights gained from analyzing the FIA Hawaii data along with a focus on soil carbon spatial drivers and distributions. And I would first like to acknowledge the contributions from my co-authors, Dr. Susan Crow and Aaron Betcher, as well as funding from the Forest Service and USDA Partnership for Climate Smart Commodities. So I think all of us here know soils are complex systems, and we often use mean estimates to characterize their properties and behavior, but those means often poorly represent the soil distributions across space and time. Hawaii's unique and diverse ecologies represent both sinks and sources of carbon under different management scenarios, but we don't have a quality baseline map of soil carbon stocks across the main islands. And current carbon and climate assessment frameworks only focus on above ground carbon, but an estimated 71% of terrestrial carbon in Hawaii's ecosystems is soil organic carbon. So these improved data-backed estimates are crucial for understanding current and future climate change and also land management impacts. So here are the approximate locations of the FIA plots across the main island chain. And of those, we have about 215 plots that have been soil sampled in 20 centimeter depth increments down to either a meter or the restrictive layer. So that's the difference between Hawaii soils and mainland soil sampling in the FIA scheme. And so just as a quick teaser, I plotted soil carbon stock in megagrams per hectare by island with the means in red. And you can see Lanai's lower mean reflects very low sample numbers, but other than that, you can see that otherwise similar means across islands don't tell the full story of the carbon distributions themselves. So I'll be digging into that aspect throughout the rest of the talk. So exploring the relationships between plot level soil carbon and above ground biomass data, which is often a stronger relationship in temperate systems. So now we're going to be looking at why that's not the case here in Hawaii with that 0.01 R squared between AGB and soil carbon. And just before answering that question, I did want to check for correlations between any other plot level FIA data that might be expected to explain some of the variation in soil carbon. But as you can see, when you look at the soil carbon, there's nothing beyond the expected strong correlation between soil carbon and soil nitrogen. And just zooming in to make that point, we don't see the typical positive correlations with biomass, environmental, or topographic variables. So I'll spend the next couple slides digging into why we think that's the case. So you can see the gradients in parent material age from northwest to southeast across the main island chain. And you've got the most recent lava flow in September of this year on Kilauea. So these are still forming young volcanics on the big island of Hawaii, all the way to 5.2 million year old weathered parent material on Ni'ihau. Thinking about climate data and climate drivers, especially the drastic rainfall gradients on the Big Island, we can see how they manifest as distinct moisture zones on the windward and leeward sides of the islands, and really look at different distributions that could be associated with kind of the moisture zones that are typical of, say, Hilo area on Big Island, super high rainfall, and then very dry areas on the leeward sides. And then we have kind of the 2017 USGS baseline carbon assessment of Hawaii, we can start to see some correlations between the parent material and rainfall gradients and the annual gross primary production of the early 2000s. So thinking about kind of how some of those drivers combine to produce these distributions, which will then influence soil carbon stocks. So this is how NRCS, the Natural Resources Conservation Service, maps the soil suborders across the Big Island of Hawaii. And soil suborders are reflective of the climate variables, including temperature, moisture regime, and the underlying parent material and mineralogy, in addition to other key features. And of course, we can see the enormous diversity of soil types across Big Island, and then the enormous diversity across the other main islands as well. So given all of these significant drivers and variation in the drivers across the Hawaiian Islands, how can we start to build models that are better representative of the diversity across spatial scales and get a better handle on the carbon distributions across landscapes? So this gets into some of the data-driven approaches I've been developing with my co-authors, where we're leveraging additional data sets and GIS data along with the FIA soil data, and using random forests and also generative approaches like convolutional neural networks to generate soil carbon and covariate distributions. And we do that to answer questions like, what are the significant drivers of soil carbon distributions and how do they spatially manifest across Hawaii's forests? So this is one result from an initial random forest approach we use to examine different covariate and soil carbon distributions built from synthetic training data. And in short, we used kind of a fixed radius around the fuzzed FIA plot location, and then randomly shuffled the association between soil carbon and a suite of climate, biological, and physical covariates to build soil carbon distributions that we then grouped into quantiles. And that allows us to assess the relative proportion of soil carbon drivers associated with different carbon distributions. So the outlined regions are broadly contiguous mapped soil suborders that I used for regional groupings. And I just here pulled out one example of one of the soil kind of mapped suborder areas on the Hamakua coast up here, northwest Hawaii Island. And we can see from the pie chart distributions here that climate is the predominant driver of the low to moderate carbon stocks, and that we have kind of an equal proportion of biological, physical, and climate drivers influencing the higher carbon regions and those higher distributions of soil carbon. So this is one promising approach for looking at regional scale drivers and thinking about them as both as distributions instead of as mean estimates. And now I'm just going to hop real quick into results from a different approach using convolutional neural networks, but the same data sources and similar deep learning. Well, this is a deep learning approach to generate quantiles, which is similar to in the random forest method, this time just using deep learning to get soil carbon predictions and just to show the success of measured versus predicted for one example quantile in this approach. This is the median. And then when we map that, we can examine regions of lower and higher soil carbon, and in this case, we're not predicting soil carbon stocks. This is just predicting the 0 to 20 soil carbon percent. But as we continue compiling soil carbon stock data, we will then be able to use the same method to generate soil carbon stock predictions. And so if we look a little bit further into the spatial patterning that we see here of the high and low soil carbon in this northwest Hawaii Island region, we can see that intensive pineapple and sugarcane monocropping, which is that historical intensive land use category here, that happened in the early 1900s and depleted soils of nutrients and carbon, and that's really evident. It jumps out here in the regions of low soil carbon across the islands. Despite some of those regions being currently high above ground biomass, we do have eucalyptus plantations all along here on the Hamakua coast, but they're still low soil carbon. So just thinking visually about some of those things that are driving higher and lower regions of soil carbon. We can also return to those soil suborders that I mentioned as broadly representative of climate and mineralogy, and visually assess if the spatial patterning in high and low carbon areas make sense. So just as a couple examples, these aquifers here are expected to be higher soil carbon. They're wet inceptisols with moderate development, and compare that to these cambids over here, which are dry and least developed iridosols. So we can see that there is some promise in this approach. And yeah, we also have the vitrans up here. This is closer to this is the side slopes of Mauna Kea up here, where we do expect some high soil carbon, young soils, but you know, lots of precipitation and high above ground biomass as well. So some of the takeaways at this point are the high carbon Hawaiian soils like andisols and inceptisols are actually better predicted in these novel data-driven frameworks than in traditional process-based models. The framework can capture soil carbon distributions that reflect those inherent complexities in the drivers of Hawaiian soil formation, like the history of intensive cultivation, current above ground biomass, soil age, mineralogy, weathering status. The spatial patterning of soil carbon and driver distributions can form the basis for forest and land management decisions. And finally, the improved carbon cycling models and improved baseline carbon stock estimates will enhance our ability to manage and conserve Hawaii's unique landscapes in the face of ongoing environmental change. And just some closing thoughts about the value of continued FIA soil carbon measurements are, you know, soil carbon is not static, it's dynamic. Our study methods must account for that. So these FIA plot level data enable us to enumerate the globally important subtropical carbon stocks and changes in them over time. So I would encourage agencies and stakeholders to prioritize data collection efforts that support high spatial density sampling and repeat sampling to the full 1 meter depth, as Melissa mentioned earlier. And, you know, that will enable us to fully quantify carbon balances both above and below ground in these systems and study changes over time. And one way to achieve that might be through enhancing interagency collaborations, especially between the Forest Service and the Natural Resources Conservation Service. And just as an example, the Inflation Reduction Act very recently funded the NRCS to establish a soil carbon monitoring research network across the US. And so the Forest Service should collaborate to increase the impact of the FIA soil sampling efforts through that type of collaboration. So with that, I might have time for a question, but feel free to reach out to me at my email. Thanks.
Thanks so much, Haley. That was super interesting, and thanks for your thoughts there at the end as well. We might have time for a super quick question. If anyone has one, feel free to speak up. And if not, if you have a longer question, feel free to put that in the chat as well. It looks like we have some good discussion going on there. I don't see any questions specific for you at the moment. But it is 3 o'clock now, so thanks, Haley. We will move to our next presenter. Our fourth speaker for today is Joe Edwards from the University of Tennessee, Knoxville, who will explore the drivers of decoupling between tree and soil fungal communities.
Hey everybody, my name is Joe. Thank you so much for having me here. I'm a postdoc at the University of Tennessee, and we've been using FIA soils along with some marker gene amplicon sequencing to understand how well soil fungal communities are sort of coupling with tree communities as they change over time, and kind of what determines if they are or not. And so I just want to start by acknowledging all the help we've gotten from our amazing undergrads as well as the resources at the Forest Service, Northern Research Station, and the Western Research Station, as well as the funding from the National Science Foundation and the Forest Service to do this intensive sequencing work. So, right, big surprise, global change is changing forests. Wow, I'm sure this is breaking news for all of us, right? But what does that mean? What does that mean in the context of these communities and how much they're changing over time? Well, because we can see and count trees and have been able to do so for quite a while, we have a pretty good idea of what the future of forests is going to look like above ground, right? We can build these models based on previous change and sort of predict where we would expect to see more change or less change spatially based on those above ground communities. But how do we do that for below ground communities, right? Because of the relative recency and data intensiveness of understanding soil, fungal, and microbial communities, we really don't have these long-term historical trajectories for those communities. But we know how trees are changing, and we know that soil, fungi, and trees often correlate with each other. So our assumption is that changes to above ground communities should correlate with below ground communities. And that's because of these important relationships with different trophic levels between trees and fungi, right? So litter saprobes or saprotrophs, we know can change quite a bit between, say, coniferous and deciduous forests, or based on the quality of the litter, which is a species conserved trait, as well as pathogens, right? We think of lots of different pathogens as species specific in their hosts. And then the symbionts as well, or they're mutualists, like mycorrhizal fungi, changing from arbuscular mycorrhizal hosts like maples to ectomycorrhizal hosts like conifers or hickory. So we can imagine, based on that sort of coupled trophic model, that as we see changes in the above ground community for plants, we should see similar changes below ground in terms of their overall magnitude and direction, right? But as I said before, we haven't really been able to measure these long-term patterns for below grounds. So how can we assess these? What can we do? Well, we don't have a time machine, but we do have the beautiful archives that the FIA has developed, right? And so I think we saw a slightly better map than this earlier today, but right, FIA has a lot of plots across the United States, and many of these plots have been longitudinally sampled, right? So the same plot sampled multiple times, a decade or so apart. And that means that they've saved soils from those trips, and we can kind of look at the community between time point A and time point B. And so some collaborators and I, including Grant Dumke with the Forest Service, published a paper earlier this year, basically showing that the archival methods that the FIA uses can be useful for understanding microbial-derived variation based on the environment. And it's similar between air-dried soils and frozen soils, right? And so we can use the air-dried soils that the FIA has collected and archived to kind of understand these environment microbiome interactions. And so, thanks to Grant and our awesome collaborators at the Forest Service, we were able to get access to a lot of these soil samples from these archives. As you can see, a very happy picture over here, and basically dip the spoon, as we said. We could access these soils, extract DNA from them, and then start to sequence that DNA for fungal communities to start to understand what those look like, right? And so we were able to use the previous collected forest census data, as well as the environmental data from the FIA plots, as well as the soil fungal community data that we collected and analyzed based on amplicon sequencing to sort of connect these two things, right? And so we were able to build this map of all the plots that we could find and identify that were sampled recursively, that we could get soil and extract the DNA from, right? And so roughly 400 plots here, sampled about a decade apart, essentially seeing how much the trees and the fungi are going to change between time point A and time point B, right? And so based on this idea of coupling or shared variation among trophic levels, we would expect that a small community turnover, ecological distance between time point 1 and time point 2 for trees should correlate with a small turnover for fungi. And similarly, if we see a big turnover, a big change for trees, we should see a big turnover for fungi. And this coupling effect is really nice because this assumption is built into a lot of Earth system and ecosystem models. Basically, because we don't really know what these turnover patterns look like for fungi, we often assume that they're based on trees. And so that, as you know, we've used that to predict the future of a lot of different carbon, nitrogen, and other biogeochemical cycling processes, particularly in response to global change. However, if we don't see that, relationship, right, we see a negative relationship where big change in trees is small change in fungi, or even, you know, some sort of other asynchronous or otherwise idiosyncratic relationship between trees and fungi that could indicate that they're decoupling, right? That the forces that affect the turnover of tree communities don't affect the turnover of fungal communities, and vice versa. And that could have really big implications for how we use those variables in trying to predict the future of ecosystems, right? And so based on, you know, the data that I showed earlier, we were able to assess the turnover in the tree communities, the change in time point between time points, and the change in the fungal communities. And what we found is that the fungal communities are changing a lot more, or a lot faster than the tree communities. Basically, here we're showing the Jacquard distance, the ecological distance matrix. Basically, imagine a Venn diagram, and all the part that's not shared between time point 1 and time point 2 is how big the number is, right? So a big number means high turnover. And fungal communities are turning over roughly 2.5 times as much as tree communities. But fungi and trees are different, right? Like a tree is a big organism, might not necessarily change all that much over the course of 10 years. And so we can sort of partition this overall turnover into, you know, changes in the relative abundance of tree species, or tree and fungal species, and the actual identity of these tree and fungal species. And what we see is a really big difference between those. So when the fungal community turnover was really high, and that was, you know, almost entirely driven by changes in identity, right? So substitutions of different species within these communities over time, whereas the tree communities weren't really being replaced necessarily by different species, but more based on the relative growth rates of the trees and species that were already in these plots, right? And so because the FIA, you know, is a quantitative index, we can actually measure how big these trees are between time points and use that to understand the relative growth rate, right? So fungal communities are changing more and primarily dominated by changes to species identity, whereas tree communities are changing less and more dominated by changes in the relative abundance. And so going back to the big question here, right? Are these coupled or decoupling? We imagine that a positive relationship would show coupling, but, you know, not positive might mean decoupling. And we found a real flat line. We looked at this a bunch of different ways. All these colors essentially represent the ecoregions in these environments. And basically what we found is across the board, there was almost no predictive power in the amount of change in the fungal communities based on how much the tree communities were changing, right? We really didn't see any ability, you know, among different regions, among different latitudes, thinking of a bunch of different variables and ways to condition this data, that these two processes don't seem to be very well related, or at least you can't predict one based on the other, right? So what is predicting these relationships? What can we pull out of this data? Well, the first thing we wanted to do is think about space, right? And think about latitude particularly, as we know a lot of biodiversity gradients follow latitudes. And so when we looked at the latitudinal response of you know, the overall turnover of the tree communities, we found that there was a positive relationship, right? And so that tree forests at higher latitudes are changing more overall than forests at lower latitudes. And this kind of makes sense thinking about the idea of higher latitude forests growing more based on that relative abundance. And we saw almost exactly the opposite relationship in fungal communities, right? When we look at the standardized beta value, it's almost the same and in the opposite direction as the tree communities, which means that fungal communities are changing more at lower latitudes, right? And so right off the bat, we see this directly oppositional response of these two groups, where one factor is driving a completely different relationship between these two trophic levels. And so, you know, we have a lot of data points here. We can include a lot of data in these models. And so we were basically able to use biotic and abiotic variables from the FIA and other databases, like the alpha and beta diversity of the trees, the mycorrhizal type, their basal area, the functional traits of the soil fungi, climate attributes and soil attributes, and compile a pretty big, you know, data set for each of these individual plots. And this is based on the starting conditions, because we're trying to predict how much they're going to change, right? And so if we know the conditions when they start, what variables seem to be important for these two groups, for how much they're going to change in end up, right? And so we took all these variables, stuck them into a model selection framework, and then ran variation partitioning on the significant variables that popped out. And what we found is that we could actually explain quite a bit of this variation in decadal turnover. So for trees, we got roughly 30%, and with a relatively good split among the biotic and abiotic variables. And what was interesting is that for fungi, we got over 40% of the variation. As a microbial ecologist, anytime we see an R squared of 0.4, that's really exciting for us, right? But what was interesting here is that a lot of this was taken up by the traits of those fungal communities, right? The relative abundance of these saprotrophs and pathogens and their alpha diversity and things like that. And when we think about what variables popped out here for these two groups, we saw that there were 22 shared variables out of a total of 68, right? So roughly 2/3 of the variables were independent for trees and fungi, basically saying that there's more that they don't have in common than that they do. Another interesting thing is that we sort of expected these edaphic and abiotic variables to have a strong relationship with fungi, right? Because they're living directly in the soil. But edaphic variables explain twice the amount of variation in the turnover of tree communities than in fungal communities, right? So fungal communities are a lot more sensitive to these abiotic components than trees, and trees are responding more to their environments. And we can break that apart into those different components for the balancing gradient, which means the species identity and the relative abundance, and see that there's big difference in these two, right? So not only do these communities differ in how much and what factors are influencing their change over time, the factors that are influencing differ among the different types of turnover, right? So this presents a really complicated model for predicting these things, but it goes to say that trees and fungi aren't the same, and we're not going to be able to use the same values and the same variables to predict how much they're going to change. So we need to start thinking of these communities independently. Overall fungal communities don't necessarily align with the turnover in tree communities, but based on our model, we showed that a bunch of different fungi might have more specific relationships with trees. And so we looked at mutualist pathogens, saprotrophs, and found that we can still explain quite a bit of variation for these different groups, but trees still really aren't doing the jobs. And one particularly interesting aspect is that pathogens, which we often think of as really species specific, literally 0% variation we could explain with tree traits, alpha diversity, basal area, anything like that. And so there is some decoupling. There is some really major decoupling between these two groups, such that their change over time isn't necessarily related to each other, and that this decoupling is linked to variable rates of change across these different spatial and environmental conditions, and that the unique biotic and abiotic factors associated with above and below ground turnover are independent of each other, right? We can't use a unified set of variables to predict both of these different groups. And so what I showed today was just the plots that we could find, replicated samples. Sorry. Basically, this is really important because we use this coupling model to predict the responses to ecosystem function change. And the fact that they're decoupling means that we have to consider them independently when we're predicting these functions. And so this severely complicates the way we predict these responses of ecosystems. And so what I wanted to get to was that we only use the samples that we could find paired plots over time, but we've been able to sample a lot more than this to get at spatial questions that I can't address today. And so these are all the samples that we've been able to get access to currently. However, we have more resources to do more research. So if you know of the location or identity of any samples that are outside of where these green dots are, I would really love to talk to you about potentially getting access to them to start measuring some of these microbial conditions and aspects. Thank you very much. I would love to answer any questions that you have.
Thanks, Joe. I do think we're out of time for questions, but if anyone has any, please feel free to drop them in the chat. And we will turn to our next speaker. So the next speaker is Sean Cahoon from the Forest Service Pacific Northwest Research Station, who is presenting about fire severity and ecosystem carbon in Alaska's boreal forest.
Okay, great. Thanks. And thanks, Melissa and Ashley, for putting together a couple of great sessions here. So the work I'm going to take you through today comes from interior Alaska, looking at how fire has started to reshape the carbon pools in these forested ecosystems. So for context, the boreal forest is the largest terrestrial biome. It's also a really large pool for global for soil carbon. But it's also a place that's undergoing some pretty rapid changes in climate, some of the more dramatic increases in temperature occurring at these northern latitudes. And that's coupled with these with an increase in fire severity and fire frequency. And we know that wildfire is really integral to boreal ecology, really influences successional pathways, and many species depend on that fire disturbance to regenerate. But what we've learned over the last several decades is that fire severity really can affect the trajectory of these successional pathways, mainly through modifications to the below ground environment. So this alternative stability domains hypothesis has been kind of generated by Jill Johnstone and her crews. And what it kind of explains is when we have two different fires that come through, in a low and moderate scenario, we have sort of a regeneration of black spruce, and that's because there's very little disturbance to the organic layer, and that seed bed or that below ground condition are really beneficial for the regeneration of black spruce in that location. However, when a severe fire comes through and it consumes that entire organic layer, it reveals a mineral seedbed that really is preferential for deciduous generation and resprouting. And those are really fast-growing species. They're able to kind of dominate that stand and kind of replace that black spruce domain. What I'm interested in is examining how these changes in successional pathways affect ecosystem carbon pools. So with that kind of framework in mind, I had some pretty basic objectives here, which were just to simply quantify the effect of fire severity on the ecosystem carbon pools and examine whether these changes in fire severity have affected this change in forest composition. And then I also wanted to take a look at how a forest has recovered in these carbon pools and the forest structure following these different fire severity scars, and examine whether we're seeing this reorganization of carbon across the landscape in response to fire severity. So with that in mind, the sample that I'm playing with here comes from interior Alaska. We have it's kind of the bleeding edge of FIA. These are new installs all across interior, and they stretch from the Canadian border all the way out to the YK Delta here. And what I did is I took the over 900 FIA plots and just intersected those with these monitoring trends in burn severity polygons. Now, those only go back to 1984, but we had we had plots that fell within fire perimeters that don't have MTBS data. So I relied on some fire perimeter data from other agencies. So you can see kind of the distribution of those plots there. And I was a little surprised to see how many of these plots fell into the unburned location. But we do see a pretty good spread across the other MTBS categories there. So with that in mind, I just wanted to kind of point out that the value of the spread of these data. Much of the work from interior Alaska in the past has come from the road system, and there's very few roads. It's a very logistically hard, difficult place to work, and we have some brilliant field crews and coordinators that pull off an amazing job every year and bring us back these data. And because we now have data from almost 20 degrees of longitude and 6 degrees of latitude, it really broadens our inference when we ask these questions about how fire severity is reshaping ecosystem carbon. So what does it look like then? How has fire really affected these carbon pools? So this is a look at the below ground environment here. So the top panel here just looks at the thickness of the organic layer. And along the x-axis there, you see the different MTBS categories. And sure enough, we see a pretty substantial reduction in the thickness of that organic layer. So it's a 39% reduction there in the high severity fires compared to those unburned locations. And we see a similar trend too in soil carbon. And we tally that up across these different NTBS scars. We see that the total soil carbon, so that includes the mineral soil, that was reduced by about 16%. But most of the organic layer has been reduced in carbon content by 28%. So it's a pretty substantial reduction here in below ground carbon, and that's where most of the carbon is. You could see on the y-axis there, the scale is pretty substantial. And I also just wanted to point out here that the reduction in the organic layer that really has some knock-on effects when you start to disturb that that organic layer. It really has effects on other components of the physical system, permafrost being the main one. That thinner organic layer usually leads to warmer soils, which can disrupt the permafrost and create a deeper active layer. It can stimulate microbial activity and the long-term decomposition of these carbon pools over time, not just in that initial combustion of the organic layer. So this was sort of a nice look at how we are seeing these differences across the NTBS scars in the below ground environment. But what about the above ground? So again, we're seeing this is live trees here in the top panel, and we see a pretty stepwise reduction in live tree carbon. You would expect that with fires, but it is kind of confirming to see that we are seeing that those high severity fires are taking out most of those live trees. Now we see sort of a mirrored response here among the dead tree carbon mass, where we see actually an increase in dead tree biomass as you get into these higher severity fires. For some reason I thought it would be you wouldn't see that much of an increase in dead tree carbon mass in these high severity fires, because I just assumed those fires are super hot and they just combust the material. But I think what we're seeing here is maybe just sort of the really fine scale heterogeneity that these fires produce, that doesn't necessarily combust those trees, but kills them kind of over time. So again, this is sort of an idea to kind of really look at how these fire severity scars have affected these different carbon pools. But within the framework of the alternative stability domains, I wanted to see whether these fire severity scars were changing the composition of the above ground biomass. So what I've done here is look at the proportion of live trees per hectare in the time since fire among live deciduous trees on the left-hand side and live coniferous trees on the right-hand side with respect to the NTBS categories. And what we see here is that we see a pretty strong and rapid increase in these deciduous trees over time in these moderate and severe fires. But for the live coniferous trees, we see that they kind of dominate these low severity fires, and this is helpful. It really kind of confirms what we might predict from the if we know something about the alternative stability domains hypothesis, that under these high severity fires, we would expect live deciduous trees to really replace the conifers and kind of dominate these stands within a few years of the fire. Another way to look at this is to look at the seedling data. And so here I've just graphed the number of seedlings that are coniferous and deciduous for each NTBS category. And as you might expect in these unburned stands, you see a pretty high proportion of those seedlings are coniferous, but they start to kind of level out a little bit as you get over to these low, moderate, and high severity fires, kind of representing the encroachment of those deciduous seedlings kind of taking hold in these burn scars. However, I would also point out that that we are seeing actually a pretty substantial contribution from these coniferous seedlings in these high and moderate stands. And I think it's worth kind of remembering that when we want to think about sort of very long-term trajectories of how these stands might recover following fire. I don't think it's quite as simple as just there's now a high proportion of live deciduous trees when we have this kind of lingering seed bed of coniferous species kind of hanging out in the understory. Okay, So now that was kind of a way to kind of look at how the fire severity has affected the composition of these species, of these stands. But we can also look at how carbon has recovered from here. Now, if we heard this morning about the dangers of space for time substitution. So Margaret, maybe just cover your ears if you're listening here. But the point that I'm trying to make here, though, is just that you see these different carbon pools in each panel here, and what I found is that there's a lot of scatter and it's not super obvious, but there is a pretty strong and significant increase in carbon pools in these live deciduous trees that we don't see in coniferous species or in the carbon pools. Now, the visuals aren't great, but the statistics hold up here. You can see the coefficient of the slope here in the table. Now, I just want to point out, though, that this is a 30-year snapshot in time of live coniferous trees and the recovery of soil carbon. And we know that these coniferous trees can live for centuries, and soil carbon obviously builds up for centuries as well. So I'm not terribly surprised that we're not seeing a strong response in these 30-year windows, but maybe over longer periods of time you would. Okay, so I just want to kind of wrap it all together here, and hopefully trying to paint a picture here of how fire severity has altered the ecosystem carbon contribution here. What I'm really trying to get at here, hopefully trying to illustrate for you, is that the forest, when it burns, the recovery of carbon following fire really depends on the severity of that fire. So if we start with a scenario where we have these initial conditions, where we have a mature, unburned black spruce stand with a large soil carbon pool, then we have a fire that comes through. It can be low, moderate, high, and obviously anywhere in between there. And we see a pretty substantial reduction here in total carbon, as well as live trees and the soil organic layer. And we know that under the alternative stability domains, and from the evidence we saw in a few slides ago, that these low severity fires are actually kind of they seem to be returning to this conifer dominated stand. And so what I've done here is just taken sort of these older stands that we know burned, and they burned between 1951 and 1983, but they are now classified as coniferous. They have more coniferous trees on the plot than there are deciduous, was my simple way of classifying it. And so when we look at these carbon pools here as an analog for what we might expect under these low severity fires, we do see that there's a reduction in ecosystem carbon, and there is a loss of carbon in the live trees compared to these unburned coniferous stands. However, I would caution, though, that this is, again, a short snapshot in a very long-lived system. On the other hand, when we have these high and moderate fires that come through, we're starting to see this transition towards deciduous dominance. So these are fires that burned, again, 70 to 40 years ago, and are now represented by a total increase of about 10% in ecosystem carbon, mainly driven by live trees. And that live tree proportion is mainly driven by this increase in deciduous trees. Again, it's a short snapshot in time, and analog is not necessarily, or the past is not necessarily an analog to the future, but it does give us a framework to work with for any deviations from these means or deviations from what we might expect as we get to remeasurement period, hopefully. Okay, a couple of concluding points here. So again, my goal here was to sort of meld the MTBS data and our new FIA data to address this the alternative stability domains question, and whether there is a change in stand composition and ecosystem carbon. And so we are seeing that there is these high and moderate fires really reduce carbon in both the below ground and above ground. But the return to or the transition to deciduous dominance likely is kind of recovering a lot of that carbon lost in the above ground pools. However, we do know that deciduous are not invincible. They are vulnerable to pests and disease. They're not as they're quite climate sensitive, and they're short-lived. And that short-lived component is important here when we also kind of return to that consideration of the high presence of conifer seedlings and suggest that long-term recovery. We also are seeing this kind of northward migration of spruce bark beetle that might play into this as well. So all I would say there at the end is just that the persistence of these above ground carbon gains following severe fires is still unclear in the long term. I just want to thank you guys for a great session and your attention today, and our great field crews and the soils lab that process our data. Thank you.
Thanks so much, Sean. If there are any questions for Sean, feel free to drop those into the chat. Our final speaker of this session is Colby Brungard from New Mexico State University, who will present about soil spectroscopy for the measurement of soil properties on RMRS-FIA plots.
My name is Colby Brungard. I'm giving this presentation on behalf of a senior researcher, Kanchan Grover, and I, who are both at New Mexico State University, and John Shaw, who's a research forester with the Rocky Mountain Research Station. And we're looking at the ability to estimate and measure soil properties on all Rocky Mountain Research Station FIA plots. The FIA soil collections in the Western state has been more or less ongoing continuously since 2000, and the states in this are listed there. In some of the states, inventory actually preceded the annual inventory. So we have some soil collections from quite a long time ago. At the end of 2025, about 14 of the forested plots will have been visited and have soil data by the end of next year's field season. In addition, the Pacific Northwest FIA program has collected soil data in the states listed there over the time frames. And all of this data, pardon me, all of the soil samples have been collected and returned to the Rocky Mountain Research Station and archived. And here is just a map that John has prepared showing showing the spatial distribution of most of where we have samples. There's still an additional samples that are not shown here. As we've been digging through the archive and associated databases, we've discovered other samples that exist that don't exist in the archive. And so this has been, as part of this project, we've also done a lot of legacy data capture and rescue. Because of all the investment that the Rocky Mountain Research Station has put into collecting these samples, really there's a lot of interest in improving and leveraging this archive to improve the soil data value for all of the Western samples. So as part of this large project, almost all of the samples in the archive have been shipped to NMSU, to New Mexico State University. We've invested a lot of time in organizing and labeling and building a database and then subsequently analyzing these samples, these archive samples. And so currently we're analyzing them for soil texture. Currently, as has been mentioned, soil texture data in the FIA database is simply just 4 classes. We're using laser particle size analysis to improve the characterization, or at least the representation of these samples. So this is an example of plotting some of that data, what it looks like. The x-axis here are just size classes, and I've just labeled these figures as sand, silt, and clay. And this is an example of what some of that soil texture data looks like once it's been processed through the laser. We're also measuring soil moisture capacity. In particular, we're looking at the water content at permanent wilting point to see if moisture holding capacity explains patterns in tree mortality. And we're using a dew point tensiometer to measure these for these measurements. But today, and the point of this presentation is really to talk about laboratory soil spectral analysis. And the idea behind soil spectral analysis is that by using soil spectroscopy, it's possible to measure how light interacts with soil at different wavelengths. And I've listed here some of the different tools, the different instruments that we have available. So looking at middle infrared reflectance, the reflectance of soils in the middle infrared portion of the electromagnetic spectrum. We also have energy dispersive X-ray fluorescence, laser-induced breakdown emission, and each one of these tools gives us a spectral signature that's based on basically the mineral and organic matter composition of each of the soils, and kind of, in a sense, creates a spectral fingerprint. And then the idea behind this is that from these spectral fingerprints, from individual soil spectra, it's potentially possible to estimate many soil properties for a fraction of the time and cost of traditional analysis, which means that we could replace or certainly at least supplement traditional analysis, and with the ability, the possibility to expand measurement of soil properties on all RMRS-FIA plots. One caveat to this is that this technique may not be appropriate if very high levels of precision are needed. And so the idea behind this is of how it works, and I'm just going to today be talking about MIR, or middle infrared reflectance spectroscopy. Any of these spectral techniques, mid-IR included, doesn't directly measure soil properties. Instead, we have to have a large database like FIA of known soil properties. And then by measuring the reflectance, it's possible to derive a calibration model between the spectral reflectance and the laboratory measured properties. And assuming those calibrations are robust, it then becomes possible to measure unknown samples based on their MIR spectra. And because collecting spectra is much faster than traditional analysis, this opens up the possibility of expanding sampling efforts. What this looks like in this diagram basically is kind of just what I've verbally summarized, but I like the picture, is that it's possible to take the archived soil samples, and from the archived database associated with those soil samples, we can get traditional lab measurements. And then because we have the archived physical samples, it's possible to measure the spectra. And together, the measured spectra and the database of spectral property, or I'm sorry, the database of analytical properties, combined into what we refer to as a spectral library. And on top of that spectral library, then it's possible to derive a relationship between the soil spectra and the soil property using some sort of calibration model. And I've drawn the picture here as a linear relationship. In practice, these relationships are never linear. But hopefully this communicates the idea that once then there's a calibration model, it's possible to predict new unmeasured soil samples. And the instrument that we've been using to collect middle infrared spectra is this Brungard InvenioR. It's a middle infrared diffuse reflectance infrared Fourier transform method, or MIR drift model. And it has a high throughput attachment. So we're able to collect 98 samples in about an hour and a half to collect the reflectance. We replicate many of these samples, so it's not 98 samples, individual samples. We're able to get about 20 samples in an hour and a half replicated multiple times. And the soils preceding this, the soils are finely ground. Once the soil sample goes through that instrument, it returns a sample, or pardon me, it returns a spectrum that looks something like this, with the absorbance peaks reflective of different atomic and molecular bonds. The reference for this figure is shown below. So we wanted to present today just the results from a quick initial test to see how well this would work on FIA soil samples. So we took 80 FIA soil samples from 2003, some from Arizona, California, and a few from Oregon. This just happened to be among the first batch of soil samples that we were processing. These were all mineral samples, mineral 1 and mineral 2 soil samples from the FIA database. And we then put these through the Brungard InvenioR instrument, the picture of which I just showed, to measure the middle infrared spectra. And then because this was a very quick test, rather than developing a calibration model myself, I used the open soil spectral library to develop a calibration model. So this is a very robust spectral model, a calibration model built upon a very robust spectral data set, except that these spectra were measured on mostly agricultural soils from the NRCS Soil Survey Program. But the Open Soil Spectral Library is an online program that makes it very simple to collect spectra, pardon me, upload them through a web API, and get results back very quickly. And in the background, what's happening is there's a cubist model that's a tree-based model that gives returns predictions based only on the spectra. So then we compared the actual analytical laboratory results from the FI program with what we got back from the Open Soil Spectral Library. It's just a very quick test. And the FI database has a large number of soil properties, but for this, we just showed organic carbon, inorganic carbon, and total nitrogen for this test. So here are the results, and I would like you to focus just on that middle figure for a moment. So this is organic carbon. The x-axis are the actual measured values, and the y-axis represent what was predicted from the soil spectroscopy. And each one of those dots has error bars, although you can see a few of those dots, 3 in particular, maybe 4, have very large error bars. And if you just look on the x-axis first, if you look at the measured properties, it looks like at least 2 or maybe 3 or 4 of these, although they were called mineral soils, may actually have been forest floor soils. There's 10% organic carbon and above, to me suggests a difference between a mineral and perhaps an organic soil. But if you look then at the table on the left, there's an R squared, that's the correlation. Then the LCC is Lin's concordance correlation coefficient, which basically measures the fidelity to one-to-one line. And so higher numbers are better. And then the bias is just sort of on average how different were the measured and the predicted values. And for a very quick test, this suggests that for organic carbon, this works pretty well. The large error bars, these are samples that likely are not represented in the spectral library, and these would be samples that would need traditional analysis had these been new samples. So this gives us a way to maybe prioritize which samples should receive additional analytical values or analytical measurement. If now you look at the plot on the right, this is again a measured versus predicted, but in this case it's inorganic carbon. And these results suggest that soil spectroscopy for inorganic carbon using the data, the spectral library that I had quick access to, is insufficient. And that's rather surprising. The Kellogg Soil Survey Lab is actually thinking about replacing traditional inorganic carbon measurements with middle infrared spectra because it typically is more accurate. But this suggests to me that at least for forest soils, it would be important to have FIA specific calibration built for some of these properties. And then looking at total nitrogen, again, this is a similar measured versus predicted. It looks for low values of nitrogen as well as for even some of the higher values of nitrogen. This is in percent. It doesn't fit quite as well as organic carbon, but it's still a fairly robust predictor with high R squared values, high LCC concordance values, and relatively low bias. So in conclusion, this work builds upon the 2 decades of FIA collection and analysis that the Rocky Mountain Research Station has done and archived those samples. This quick initial, this is a pretty quick initial test. We have about 500 spectra. We just randomly chose 80 that we had quick access to. But it does seem promising for carbon and nitrogen, and we'll continue to investigate some of the other soil properties. This initial test does suggest that it'll probably need a FIA-specific spectral library, which will further increase the accuracy of inorganic carbon, as well as likely the other soil properties, organic carbon and total nitrogen included. And we're cautiously optimistic about the possibility to expand this potentially to all the Rocky Mountain Research Station FIA soils and all the FIA plots, so that we could have measurements or inferred measurements, at least estimates, robust estimates at all soil, at all sampling locations. One question that I would potentially pose to the audience is how accurate do measurements need to be before we can detect change over time? So with that, thank you for your time, and take any questions.
Thanks, Colby. We are out of time, so if you have questions for Colby or thoughts on the question that he posed at the end, please drop those into the chat, or if you need a little more time to respond, you can just put those directly into the Huba chat as well for our session. So thank you all for joining, and special thanks to the speakers today. I invite everyone to join us for the second session today at 5:15 p.m. Central Time. Take care, everyone.